this session, we'll learn how to troubleshoot a routine file import issue. And once we complete that, we'll be able to find the melting temperature of our gene sequence in a program called EditSeq. EditSeq is part of a commercial bioinformatics application called DNASTAR. It has been provided to us using an educational license. EditSeq is an older DNA sequence editor, but it does have some features that are not found in other modules, including the ability to identify the melting temperature for a nucleic acid sequence, as well as the capability to proofread sequence. The proofreading capability is an artifact from when DNA sequences were manually entered into the application instead of imported from automated sequencers. A very common activity in bioinformatics is working through different issues with file formats, converting files, importing and exporting files. In this session, we'll see an example where the application expects a particular extension on a file, but the current export format does not default to the expected format. So let's launch EditSeq from the DNASTAR menu. The application opens, and then we go to the File menu and choose Import. We go to the File section where we know we've saved the CFTR GenBank formatted file. But as we can see here, even by looking for the various accepted formats and choosing GenBank, it still isn't recognizing the file that we saved. We noticed that it was looking for a .gbk formatted extension. Let's go to our folder where we saved the file, and we see here that the CFTR GenBank file has a .gb extension. So let's see what happens if we just change that to a .gbk. So we'll make that change, accept it, and then go back to EditSeq. Now when we go to File Import, we can see that now it recognizes the file. We select it and open it, and now EditSeq has brought in the file. One feature of EditSeq is that it allows us to view the melting temp of a nucleic acid sequence. We have to select all and then go to the Goodies menu, choose Statistics, and then we can see, if we scroll down, it shows the melting temperature for the CFTR GenBank sequence. Another feature of EditSeq is the ability to have the application proofread a sequence. While there's not much use for this anymore, it is kind of a fun feature.
G T C T T T G G.
Well, I think that's enough of that. In this session, we learned how to troubleshoot a file import issue by changing the extension to the imported file. We also learned how to use EditSeq to find the melting temperature of a nucleic acid sequence.